The ideal habitat for a wild honeybee colony is exactly what we see here, a dark, enclosed tree cavity of roughly 40 liters. Inside that cavity, the rough interior bark provides a crucial mechanical advantage. It gives the bees a textured surface to anchor a thick propolis envelope, a resinous coating that acts as a highly effective external immune system for the colony. Modern apiaries abandon this biology. Look at standard Langstroth box components. They are built entirely with smooth, planed wood surfaces. These boxes feature highly exposed entrances and minimal insulation, creating an environment that sharply contrasts with a tree hollow's thermal stability and immune defense. This architecture optimizes for human convenience, allowing for rapid inspection and honey extraction, but it deprives the colony of its biophysical defenses. Without rough walls, a propolis envelope cannot effectively adhere. Without a sheltered entrance, temperature regulation breaks down. And without a mechanism for natural water absorption, internal moisture simply slides off, creating a damp environment ripe for disease. By substituting a living, defensive microecology with a sterile, smooth void, standard hives actively immunocompromise their inhabitants. This structural deficit can be reversed. The Hollow-Hive v3.0 base kit is a modular hardware stack designed to rebuild tree hollow physics within that standard box. First, 4 textured liner panels snap directly into the interior walls, providing the missing mechanical grip. Next, to manage moisture and hydration, a slow drip rooflet and a perimeter bead rail are added to the top and edges of the structure. Finally, to block environmental boundary pollutants, a specialized filter cassette and membrane gate secure the entrance. None of these modules require changing the standard beekeeping frames. They work entirely by retrofitting the microenvironment surrounding the bees. The result is a standardized, scalable kit that engineers the immunological and thermodynamic advantages of a wild tree hollow back into the commercial apiary. A smooth wood surface is a physical dead end. It prevents the formation of stable microbial films, leaving the colony exposed. The v3.0 inner liner replaces that surface with 4 removable, food-safe panels made of a cellulose-lignin blend. The critical engineering detail here is the geometry. The panels feature microfissures precisely sized between 0.8 and 1.2 millimeters. That specific tolerance provides the exact mechanical grip required for bees to deposit and maintain a thick, stable propolis envelope against the walls. Once the physical scaffolding is in place, the system introduces a secondary biological intervention, seeding the interior surface with beneficial microbes. This is achieved using sealed, deproteinized local pollen tablets. Upon installation, they release benign surface microbiomes directly into the hive. This process adheres to strict biosafety rules. Every tablet undergoes qPCR pre-screening, ensuring absolutely no live pathogens are introduced into the local ecosystem. By combining mechanical wall roughness with local microbial seeding, the hardware turns a passive, vulnerable box into an active host for a protective surface community. There is another overlooked dynamic in wild tree hollows. The interior gets wet. In nature, rain runs down a tree trunk in a process called stem flow, carrying microbes and vital solutes directly into the cavity. This diagram outlines the biomimetic solution, the slow drip rooflet. It routes a small amount of water downward, deliberately bypassing the central brood area to keep the core dry. Operating constraints here are tight. The system drips at roughly 1 milliliter per hour, and it runs strictly for the first 2 to 3 startup nights. Following the water path downward, the droplets hit the perimeter bead rail, flowing along a precise 1 to 2 degree slope towards small collection cups. Made of terracotta or cellulose, this bead rail harvests ambient dew and fog, maintaining the thin water films required by the wall microbiomes. Those same collection cups provide beekeepers with a safe, accessible liquid sampling point every month. This transient hydration mimics natural stem flow, safely accelerating colony startup without risking the severe mold outbreaks that plague standard damp boxes. Moving down to the entrance, the hive faces severe external threats, specifically from airborne particulate matter and pesticide drift. We start with an ultra-matte entrance baffle. Reducing visual glare calms the incoming traffic and significantly lowers visits from nuisance wasps. Behind that baffle sits the plastic-free boundary filter cassette and a 3-layer membrane gate. This diagram models the air intake, holding at a steady velocity of 0.5 meters per second. The boundary filter itself is composed of terracotta foam paired with a biochar and hemp pad. It is engineered to a strict constraint. The pressure drop must remain under 2 pascals. That exact pressure threshold is required to trap incoming dust and particulates without suffocating the colony or causing traffic jams at the entrance. Returning foragers then pass through the 3-layer membrane gate. The gate consists of a cellulose comb, a hemp slip treated with microbiochar, and a chitosan mesh, keeping the internal pressure drop between 3 and 5 pascals. As the bees walk through, the membrane acts as a physical brush, cleaning the foragers and capturing environmental residues directly on the hemp slip. Through precise fluid dynamics and targeted filtration, the entrance is upgraded from a simple hole in a box into an active protective barrier. Because the boundary filter and membrane gate constantly capture physical material from the surrounding landscape, deploying Hollow-Hives turns an entire apiary into a network of active environmental monitors. This data is managed through an open NRV protocol: measurement, reporting, and verification. Every month, the filter slips are removed. This diagram tracks their path. They are weighed wet and dry, and then passed through FTIR or micro-Raman lab analyses. The lab identifies the exact mass of captured microplastics, breaking down pollutants like PET plastics or tire wear surrogates. This raw data is then published as standard CSV or JSON files. By utilizing open data formats, municipal and environmental agencies can plug directly into the dataset, effectively using the bee's natural foraging radius as a regional sampling tool. The hardware stack allows vulnerable agricultural livestock to double as sensitive edge sensors for local air quality. To prove these biomimetic interventions work in the field, they are evaluated under a rigorous 6-month ABC pilot protocol. This matrix shows the testing baseline. Hives are randomized into site blocks, comparing the Hollow-Hive v3.0 against a Langstroth control. All lab testing is completely blinded. Internal sensors continuously track colony mass, temperature, and median CO2 and humidity cycles, aiming to hit specific pass-fail metrics, a 70% reduction in pathogen markers, and keeping winter mass loss at or below 15%. The pathogen reduction is verified using targeted qPCR panels, specifically scanning for aggressive threats like Painobacillus and Nosema. Beyond survival rates, these physical upgrades offer a direct economic return. By reducing disease treatment costs and stabilizing honey yields, the hardware pays for itself in just 1.3 to 3.0 years. By engineering wild physics back into the apiary and measuring it relentlessly, Hollow-Hive v3.0 establishes a biological architecture that protects the colony and ensures the entire system remains economically self-sustaining.